All right. Good morning, everybody. How was your weekend? Pretty good? Decent? Did we rest a little bit? Kinda. All the head nods went silent. Okay. Okay. All right. We're getting into one quick thing that I see here is our class attendance on Mondays is low. So try and come to class on Mondays if you're not doing it. This is a good time to really make that final push towards the end of the semester. I know they're long weekends, but definitely try and get out. I hope we're having some fun. So a couple things before we get started. There's some laughter in the room. Okay, but anyway, before we get started, I wanted to mention a couple things about exam 3. It was officially posted. You now have about a week to hand it in. I sent a message over the weekend saying how I wanted to do it was you guys can go ahead and print it off on your own or put it on your iPad or whatever it is you do, but then find a way just to scan it to me when you're going to hand it in. It's easier for me just to get them all via my email, download it to my iPad, and then grade them and send them back out to you. If there's any concerns with doing that, feel free to let me know. As for the content of the exam, I want to remind you again, you can use really anything with the exception of primary literature articles as well as yourselves. You can't obviously work on it together. That's not a good idea. And we caught that a couple times last year. So let's not have that happen. The last question, already had a couple questions on that one. It does say 2 pages that are 1.5 spaced. Just for clarification, in the past we've had people try to measure that they get to exactly 2 pages. That's not what I'm aiming for you to do. Just give me a good explanation in 2 what I would say really strong, 2 to 3 really strong paragraphs that tell me, about the paper, and then also what you got out of it. I really want to get your analysis on what you thought about that. That is your last sort of 50 points of the exam. And remember, it's worth the 50 points. So, do some nice work. I do read all of them. And I don't just put 50 on. So it's definitely worth doing. I do want to get your insight on those things. Are there any sort of general questions before we get into what will be the sort of the final section? Okay. Just to give you a bit of a heads up, it feels weird, right? That you're heading into the final section of content and that you have sort of a month-ish left. But I want to remind you that we're going to cover the anti-cancer section between today and Wednesday. And then on Friday, I'm giving you that hour to spend looking at your exam, because I know usually don't think about it till a little later. And then after that, we're really in sort of presentations from med chemists that are going to visit us, presentations from the honors folks, as well as the global health projects. Don't snooze on those. So what I would say is that there are probably going to come up a couple of questions affiliated with, in particular, your peers' honors projects. I may just sort of bring up, a question or 2 on your final. So definitely come, give your attention to them. Definitely attend class on those days to sort of support them, cheer them on, etc. I am going to plan on, as we move closer to the final, we're going to have a lot of review sessions. So those are going to be happening maybe not next week, but starting the following week. Abesh and I are going to start doing some review sessions for people. Okay. I'm going to head over to the anti-cancers PowerPoint now. Share screen. Yeah, so getting into sort of our sort of case study 3 and being anti-cancers, I start with this whole section with this molecule Taxol. I'm sure how many of you actually heard of this molecule? A few? Oh, that's interesting. Okay. So there's a reason why I started this. I think when I think about sort of the anti-cancer section and anti-cancer in general, I would say that this is maybe one of the most inspiring areas for medicinal chemistry overall. And a lot of that actually started with this molecule in particular. It was discovered by this plant called the yew tree, and it was isolated, and it was found to have all these like really wonderful anti-cancer properties. Right now, it's used sort of across many different cancers, so like lung cancer, ovarian cancer, breast cancer, etc. And usually it's used as sort of a last resort in that it's so potent. But at the time, sort of in this discovery in 1960s, 1970s, it was actually really remarkable in that you had this sort of really complex molecule that was so potent, right? It was one of the most potent anti-cancer agents ever discovered. But it sort of initiated all sorts of different studies in biochemistry, chemical biology, organic synthesis, et cetera, because of how complex of a molecule was. And so if you ever talk to anyone that's really intrigued by the whole area of organic chemistry, this is one of the first major tests for synthetic organic chemists, right? So you've seen throughout our conversations a lot of sort of flat molecules, right? A lot of molecules that have sort of aromatics affiliated with them, or they're sort of multi-aromatic type species. So when you look at this molecule, right, it has a ton of stereochemistry affiliated with it. And so that's intriguing for many different reasons. One from sort of the synthetic standpoint, but then also you have to imagine like the binding of this, or the understanding of the binding of this molecule is really, really complex. And so this is actually one of the major challenges. So they wanted to produce a whole bunch of it. And so at the time, the thought process of even pursuing this molecule was like crazy, or you didn't have methods to do that. And so just to give you some numbers, Taxol was the topic of sort of intense research by over 30 research teams in the United States in the 1970s. The first total synthesis of this molecule, right, to generate enough, right, so then you actually generate a fistful of it so that you can administer it to patients, was not done until 1992. And even at that time, when they made it, they made like a milligram of it. So even from, let's say, 1992 till early 2000s, there really wasn't enough of this molecule to even be a sort of a viable treatment for people. And on top of that, during that time, they were isolating it, right, from the tree, and they're not getting a ton out of it, right? So there's been, I would say, even to date, efforts on this molecule are still ongoing. And I would say this is probably one of the most funded molecule topics that you can find in the world. So if you ever get an opportunity to read into a little bit of the history of Taxol, it's the stories are unbelievable. But maybe more importantly, sort of anti-cancer is arguably one of the most important areas in medicinal chemistry, and maybe one of the most intriguing areas to most. And hopefully we'll get to talk about a lot of that as we go through. Right. So cancer, I'm sure a majority of us have been affected by this at some point, whether it be ourselves, our loved ones, etc. But it definitely is, I would argue, maybe one of the most feared diseases in the modern world. The World Health Organization estimates that about a third of people have been affected by it, and approximately a quarter of all deaths in the developed world it contributed to in 2000. That number remains pretty consistent. Somewhere in the neighborhood of 10 to 15 million people die every year. And so it makes it, of course, a major topic, sort of across the medicinal chemistry field. Right. Cancer cells are formed when normal cells lose their regulatory mechanisms, controlling growth and multiplication. And we'll talk a little bit about that as we go. Before we get into the discussion, differentiation, a few definitions that you do want to know, because they'll be brought up a few times. One of them being differentiation. So the actual distinguishment between types of cells. The neoplasm, so new growth, you won't see that one as often, but I want you to at least have that. Cancer or tumor is something that we'll be bringing up, often, more commonly accepted, and will be used throughout our discussion. And then benign, cancer that's localized, so one that's not spreading. And then malignant, if the cancer cells invade other parts of the body and are able to set up their sort of secondary tumors. That's called metastasis. And so what causes cancer, right? I'm sure you've seen many different sort of advertisements about this and how we go about preventing it, et cetera. And of course, the tobacco industry has been one that's hit major, right, in this area. And so, of course, smoking, right, has contributed to a ton of different types of cancer. You could see, right, effects pretty much everywhere in the body. And it's actually, even some of the efforts, right, that have been used to sort of stop advertisements for cigarettes and things of that nature, it's still, of course, a prevalent area. And so, what's really sort of causing the problems, and usually it's sort of they're sort of referred to as like poisons that you're exposed to when you, for example, when you smoke a cigarette. And usually those are sort of nitric oxides, reactive oxygen species, et cetera, that start to tear down your immune system. And it doesn't bounce back, right, from that sort of sustained stress on your body. And so, I think this is a really good, example of, we think about, generally speaking, when you think about disease, you think, maybe it's people that aren't necessarily taking care of themselves, et cetera. But I think the way I sort of think about it is, how much stress are you sort of putting on your body, right? So you put stress on your body by of course, tobacco use, put stress on your body by, potentially, being overweight, et cetera. And we'll talk about that in just a second. But then on top of that, and something that I don't think we think about a lot, especially for, the individuals that are in this room and on Zoom, is the stress that you put on yourself, throughout the semester, your job, et cetera. And you get busier over time. And so, I think this is maybe another good reminder to make sure you're taking care of yourselves throughout the rest of the semester, and of course, the rest of your lives. And so, so there's 13 cancers, right, that are affiliated with being overweight or obese. As you can see, very similar to the tobacco problem, right? It can sort of affect all areas of the body. And so, what have we started to do about that? A lot of it has been related to sort of advertisements about one, sort of the negative effects of sitting still all day or smoking cigarettes, et cetera. And sort of depending on the community that you're talking about, there's been sort of maybe some key aspects to get thinking about. So, in schools and early childhood environments, right, they sort of encourage walking to school or biking to school type programs, support sort of quality physical education, increasing healthy food options. And now, actually, in many schools are taking out a lot of the, I would say, poorer food options for students. In the workplace, and definitely, this becomes more and more apparent, especially for people with desk jobs where you're doing a lot of typing throughout the day or phone calls, et cetera. Definitely the encouragement to get up and move around every so often, using the stairs instead of the elevator, are a couple good examples. I mean, the one sort of in the bottom left, sort of at home that it was so interesting. I was looking through these slides, again, getting ready for this, and it popped right up on my phone that like my screen time was down by like a certain percentage last last week. And each week I kind of like monitor that and I say, okay, was it up or down? And then I was thinking about it a little deeper. I was like, oh my gosh, even if it's down 13% and you're spending still 4 hours a day or something on your screen, I'm thinking about how much of that time is really spent, sort of just, kind of just sitting down, hanging out, flipping through the phone, maybe doing the TikTok type thing that you have to do. But anyway, but I think that's always sort of inspiring to me, is to like, we definitely got to get up and get moving and do that sort of thing. And then sort of on sort of the residential side of things, right, increasing access to walking trails, right, those become more and more prevalent over time. Definitely when I was living in the Northeast during the winter time, the walking trails and the biking trails weren't too big of a deal because you couldn't get to them. So there were sort of indoor programs to actually sort of facilitate sort of a healthy lifestyle. But nevertheless, Arizona, of course, is on top of that. Of course, we're getting into the hot season now, but I always think this is pretty cool. Again, these are through the CDC, and really worth. I mean, even in the workplace that you're maybe working, worth hanging up. I think it's definitely hanging up in Biodesign C. But anyway, so to get into our a little bit of our discussion, sort of what exactly is causing cancer, and a lot of that comes down to sort of genetic faults that eventually sort of lead to cancer. So you sort of have the activation of these proto-oncogenes. These are the ones that normally code for proteins involved in control of cell division and differentiation. Those are mutated, right, and they start to malfunction. And then also, maybe the area that's a little less appreciated, right, is the inactivation of tumor suppression genes or anti-oncogenes. So the ones that are actually, giving some indication as to whether or not, that growth should continue to happen, or they do sort of the checking the repair, and sort of cause apoptosis, which we'll talk about soon. And the consequences of those genetic defects are seen sort of at the bottom of the slide. Things like abnormal signaling pathways, that'll be at the core of a lot of what we talk about. Insensitivity to growth inhibitory signals, abnormals in cell cycle regulation. We'll get to talk about that a little bit. Evasion of programmed cell death or apoptosis. So, causing sort of the regulatory processes that sort of initiate apoptosis. Limitless cell division, among others. And so, so this sort of breaks it down maybe a little more specifically between sort of the abnormal signaling pathways and the insensitivity to growth inhibitory signals. So as we might be able to appreciate from some of our previous sections, the abnormal signaling pathways are going to be a core problem. So a lot of the signals that say go ahead and initiate cell growth, they're almost sort of hijacked, right, by cancer, and they start to sort of grow and divide uncontrollably. And then the what I would say is this sort of more or less recapping what we said before, but then the key one too is that the sort of insensitivity to growth inhibitory signals is really important, such that like even though you have sort of signals that are actually giving any sort of indication that you should sort of stop the growth, there's really no sort of signal transduction in that pathway, and I'll talk about that soon. Right. So just want to remind you a little bit about, there we go. About the four phases that are involved in cell growth and multiplication. This is probably a review for a majority of you. But you sort of have this GAP1 or G1 phase where the cell is actively growing in size and prepares to copy its DNA in response to growth factors or signals. And you can follow along on sort of the bottom figure if you want to get it sort of in pictorial form. And then you have the synthesis phase where the replication of the DNA takes place. G2 phase is the interval in which the cell readies itself for cell division. And then mitosis is where the cell division actually takes place. So I think, with sort of those basic definitions of each of those phases, I think the thing to really appreciate from a medicinal chemistry standpoint is that there's going to be a ton of the system that you already have set up for cell growth and division is going to have a ton of sort of regulatory sort of spots, right? So from going from G1, for example, to S, you're going to have a whole set of proteins that sort of signal to go to the next phase. And so where that sort of comes in is in these cyclins. And so, within the cell cycle, there's numerous decision points that determine whether the cell will continue to the next phase. And then those points that we're talking about are referred to typically as restriction points. So during the G1 phase, it becomes, so those start to become a little bit irregular when you have some sort of mutation, for example. And so I think it's important to note that there's not just a checkpoint sort of after G1 and not just a checkpoint after S. There's multiple checkpoints throughout G1, for example. And so it's important to know, of course, it's a little more complex than what we sort of talk about in here. There are also many surveillance mechanisms, right? Those are the checkpoints we were talking about that assess the integrity of the process, and they can delay phases as need be. And yeah, a lot of this is regulated by cyclins, right? So these are just really just proteins that sort of bind to these cyclin-dependent kinases, and those kinases then phosphorylate other proteins that continue the cell signal. You don't have to know too much about sort of the bottom statement here, but what I would say is that where you have several different CDKs or cyclin-dependent kinases that are responsible for each of these processes. So each of the phases of cell division has several different CDKs that it relies on. And what does this sort of look like sort of from a pictorial view? You typically have some sort of external growth factor that binds to a tyrosine kinase receptor. And then you actually have, ultimately RAS is going to start the signal transduction. But you have these sort of, these are really fascinating to me. This GRB2 is sort of an adapter protein. So it's actually a protein that doesn't even have, let's say, particular binding sites, let's say. But they have more, they're almost referred to as sort of interaction sites. So even to go from tyrosine kinase receptor to RAS, that's usually how it's drawn. It's like you hit the tyrosine kinase receptor, goes directly to RAS, and signal transduction starts. But I think it's important that you note that it's a little more complex than that, in that GRB2 functions as just an adapter protein. So this brings both of them together. So they basically touch, and then the signal transduction can begin. Really, really interesting. And ultimately what that does, it produces the cyclins. The cyclins then, or ultimately our cyclins head into the nucleus, create these cyclin CDK complexes, and those initiate cell growth and division. But then you have sort of the anti-growth factors also, right? Other proteins that are going to bind to these transformation growth factors, these TGFs here, that are going to initiate their own signal transduction pathway. That pathway ultimately leads to these P15, P21, and P27 proteins that can slow down that cyclin CDK complex formation, and they do that just by binding. So binding to the spot that the cyclin typically would bind to. It's important to note that also there's a monitoring process that's also going on, such that if you know that there's some sort of damage that's occurring, you go ahead and produce P53. We'll talk about that in just a second. And ultimately, you produce P21, and then cyclin CDK complexes are slowed down again. And literally, what typically they're doing, or P21 and P53, is they're actually literally binding to the DNA and stopping sort of replication process. P53 becomes pretty important as we talk a little bit about apoptosis. Yeah, so this is actually such a cool, to me, one of the cooler processes that we have in the body. I think apoptosis, is the built-in cellular disruption process. So literally, it's like programmed cell death. Really, really interesting. So there's two distinct pathways. You have sort of the extrinsic and intrinsic route, which we'll show on the next slide. But then all the signals ultimately converge on the mitochondria containing proteins that can promote apoptosis, sort of telling the cell, it's time to get out of here. So you don't have to worry too much about bad and backs in BCL2 and BCLX. They're not going to come up in an exam or anything, but you can feel free to read a little bit more about those in detail in the Patrick book if you'd like. But sort of this is what it always looks like. So we always think about, if we want to, we always talk about, if you sort of initiate an immune response, we always refer to our white blood cells, and those go take care of business is usually what, kind of where the story stops. But I think it's important to note that, if you want to sort of initiate an apoptosis type process, right, those can actually send in what are called granzymes, and those are they're a class of like serine proteases. So they go through and they break apart proteins. And ultimately what they do is they sort of break them into parts that sort of can go into the mitochondria and sort of initiate this process that I'll refer to in just a second. But then another sort of initiation of this whole process actually starts from sort of low levels of growth factors. Right? So they're just not, there's no sort of trigger happening. And as that signal gets weaker to go ahead and initiate the signal transduction, that also sort of causes the recruitment of certain proteins to head to the mitochondria. Ultimately, what these do is they produce cytochrome C, and then what it does is it actually forms these sort of larger, complex quaternary proteins that go off and actually start to kill off cells. So actually start to perform apoptosis. The actual players throughout here are not going to be particularly important on an exam or anything, but again, Patrick will give you a little bit more detail if you're interested in reading about them. But then you also have sort of this another sort of pathway. You have these death activator proteins. It's a pretty intense name, but they bind to these tumor necrosis factor receptors, again causing signal transduction. They automatically produce caspase 8, which is also responsible for performing apoptosis. And again, right, that P53 protein ends up becoming important in sort of doing the very similar type of thing as what's happening in sort of the signals on the left. In that sort of once DNA damage has occurred, you then have P53 protein binds to the DNA, ultimately produces its own serine protease that creates BAX that heads into the mitochondria and initiates the process as well. Typically what happens is that all of these are turned on at the same time. And so it's just amazing how much sort of regulation you have throughout. Right, so telomeres are definitely key players in the immortalization process of cancer cells. Right, so this is sort of the ends of the chromosome. And so what actually happens, right, is that they're those things, every time they replicate, they lose somewhere between 50 and 100 base pairs. And so when that happens, you would think it would just sort of chew itself up, but it's actually it's been put in sort of a situation in which it can sort of it can continue the replication process multiple times, but then it's allowed to continue to do that because the cancer cells actually will produce their own telomerase. So this RNA-dependent DNA polymerase that can continue to add hexanucleotide repeats to the end of the telomere. So that's how it sort of continues to be quote unquote immortal. Yeah, so this is I'm really amazed. So the whole process of angiogenesis is so interesting to me, in that it really shows sort of how maybe hardcore cancer cells are. Right? So even if you kind of strip away all the components it needs for survival, including oxygen, it's going to find a way to get it. They're going to find a way to get it. And so I think that's really, really fascinating. So the cancer cells, right, they require a supply of amino acids, nucleic acid bases, carbohydrates, oxygen, and growth factors. So as they grow, they reach the state of hypoxia, right? So run out of oxygen, and that causes the buildup of these hypoxia-inducible factors that upregulate genes that promote survival in oxygen-starved environments. And so what the goal of those things is to do is actually ultimately produce new capillaries so that you can get oxygen to the cells for survival. So this above process, right, it's normally controlled by angiogenesis inhibitors, angiostatin and thrombospondin. Definitely, you can read a little bit more about those in the book. But then, moreover, the new endothelial cells can release interleukin-6, which then stimulates sort of further growth. Right. So as long as they know that they're okay, they have enough oxygen to continue growing, they'll do so. But they'll pause for a second and they'll produce this HIF1 if it ends up being the case they don't have enough oxygen. So, but ultimately, right before that whole process can begin, this sort of basement membrane around the blood vessels has to be broken down, which is carried out by what's called the matrix metalloproteinases. And that allows for the endothelial cells to migrate towards the tumor. And so, as you can imagine, this is not unique necessarily to the slide, but pretty much everything we talked about, each of those regulatory sort of proteins, they've all been targeted. If you look up any of the BAX, BAD, BCL2, etc., as well as sort of all of these, right? They all have had different inhibitors developed for them specifically. One of the challenges, though, is the cancer cells will also start to literally take, let's say, for example, reactive oxygen species that they're having a problem with, and they'll actually just sort of functionalize some of the inhibitors, and so that ends up being a real challenge. So actually, the design of molecules that are sensitive to sort of oxidations, for example, are a real challenge to sort of target in one place or the other. So that has a whole other story, and maybe actually Scott Bagwick from Pfizer may actually talk about that a little bit, and I'll leave that to him for a little bit. This is sort of the depiction of the angiogenesis we talked about in the previous slide, where you have sort of the growth factors, the endothelial cell, the blood vessels that are sort of allowing for sort of that expansion of the capillary network. And then you ultimately sort of break apart sort of this matrix metalloproteinases, and that's done by its own enzymes that are capable of sort of breaking through that matrix. And then ultimately what that does, it sort of releases interleukin-2 and causes sort of further growth of the cancer cells. So now as we get into sort of the agents that sort of combat cancer, right? It's going to be a lot of the very similar types of themes that you saw from before in other types of diseases, right? So you have the intercalating agents. Definitely have seen these before, right? They typically contain a planar aromatic or heteroaromatic ring that can slip into the double bond helix of the DNA, distorting the structure. Doxorubicin or these anthracyclines, huge area of study for a long time. They intercalate into the DNA, stabilize the complex between DNA and the topoisomerase II, very similar to what we've seen before. That hydroxyquinone moiety can chelate iron as well. That sort of helps stabilize the structure. And then they're also thought to interact with helicases as well. So there's a good chance that if it's affecting the topoisomerases, it's also sort of interfering a bit with helicases. So this is sort of another good example of that, right? These are essentially the sort of flat type molecules that you see, something very similar to the anthracyclines, but they don't contain that sugar moiety. Oftentimes you're going to see that trend. And the reason why you see that trend, just as an FYI, is that actually synthesis of those molecules that contain the sugar moieties are really challenging. in particular, they're really challenging to purify because they're really sticky. So it's really challenging to purify those. So start to create sort of analogs, for example, like this one on the left, that have sort of that flat structure, but then also have some polar groups that almost serve as what would be sort of the sugar type moiety out here. Non-intercalating agents that inhibit the action of topoisomerase enzymes on DNA. These ones, the camptothecins, they target the complex between DNA and topoisomerase 1. They're thought to turn on serine proteases and endonucleases. So where exactly these things bind is still not completely elucidated, right? So they're not used quite as often, but they do know that they work. So they are used in certain types of cases. Nitrogen mustards, these non-intercalating agents, are used, but they're a bit dangerous, and a lot of that is because they're really reactive. So they definitely do their job, but they don't do it in a very specific manner, right? So we talked a little bit about molecules like this. I don't know if they were necessarily nitrogen mustards, but ones that sort of have two groups that can sort of that can actually alkylate base pairs and then sort of stop replication processes. And so these are sort of several different variants that have been used. Oftentimes they mimic alpha phenylalanine, because that's one of the more common sort of amino acids to target. I think it's sort of in terms of the mechanisms that are sort of affiliated with their use. A lot of the anti-cancer agents in general usually start from prodrugs that are prodrugs in that they go through a P450 oxidation. So I was telling you before that like often it's hard to target some of the targets for I guess some of the cancer targets because oftentimes oxidation is a challenge. So what you'll do is you'll actually create a compound that will intentionally go through an oxidation process so that it can then go on and be a drug. In this case, for these cyclophosphates, they actually go through this P450 oxidation right next to the nitrogen, as many of you know, right? And then they ultimately produce this cytotoxic alkylating agent, but then they also produce acrolein. And acrolein actually, through a conjugate addition, functions as another alkylating agent. So this thing's really out to destroy when it goes out. TH302, something very similar. In this case, it's not an oxidation sort of initiates it. It's actually a reduction. So it'll go through this enzyme catalyzed reduction, probably by P450, most likely, and ultimately generate these two active alkylating agents. And the reason why they designed them this way, actually very similar to the one from before, is that they know that each of these will target a different pathway. So they know that let's say one will target like BCL2, one will target BAX, for example. And so you actually have sort of two drugs in one. It's really neat. Right, cisplatin and cisplatin analogs, huge area of discovery, drug discovery. How many of you have heard of cisplatin? Not that many, huh? No way. Okay, good. Okay, I want to give you a paper on that. That's good stuff. But it's like super cool. So it does almost very similar to what you see with sort of the intercalating agents or the non-intercalating agents, alkylating agents that you saw from the previous slide. But sort of that binding actually happens right on platinum. Though this area of development is super big. Right, then you have these CC1065 analogs. They bind to the minor groove DNA and then alkylate an adenine base. And you say, okay, well, where on this molecule is that sort of alkylation occurring here? Because it doesn't have like alkyl halides all over it. And so what's currently hypothesized is that that alkylation actually happens via a conjugate addition on one of these, and sort of those conjugate additions can happen on sort of these cyclohexanones on the left, in case you're curious. Okay, cool. Yeah, so dihydrofolate reductase inhibitors have become more and more popular. I would say in the last maybe 10, 15 years or so. You have this dihydrofolate reductase that's crucial in maintaining levels of the enzyme cofactor tetrahydrofolate. And you can kind of follow this on the bottom if you like. Without the cofactor, the synthesis of DNA building block, this thymidine building block would not be possible. So slowing down DNA synthesis and cell division ultimately. The enzyme catalyzes a reduction of the vitamin folic acid, you see on the bottom, to FH4, tetrahydrofolate, in 2 steps via dihydrofolate. And so once you have that formed, it picks up a single carbon unit to form this species that you definitely don't need to commit to memory, which is used as a 1 carbon source in the biosynthetic pathway. So the methylation of deoxyuridine monophosphate to deoxythymidine monophosphate. So that methyl is actually put on later. And so I'll get to show you that pretty soon. Just to get you a little familiar, I think as you're looking through these slides later on, maybe toggle back and forth between a few of these slides, just a few different structures. So the folic acid's up in the top, dihydrofolates on the left, tetrahydrofolates in the middle and the bottom. And then you have sort of that methylated nitrogen compound we were talking about before. But ultimately, that's where your methyl is going to come from. And so this is really cool, in terms of the design of these things, and we'll show you in just a couple slides. But methotrexate is sort of a related drug. It's one of the most widely used antimetabolites, antimetabolite being something that's going to inhibit sort of a typical enzymatic pathway. And it's used typically in cancer chemotherapy. Similar structure to natural folates, and you have to toggle back and forth just to get a feel for that. And then it prevents binding of FH2 ultimately. And so, yeah, so this is sort of the thing that typically happens. So this 5-fluorouracil inhibits the thymidylate synthase directly by acting as a prodrug for suicide inhibitor. And so some more detail sort of on how this whole process works is shown here. So typically what happens is you have a residue that'll come in off of the enzyme that'll do this sort of conjugate addition over on the left. Okay, and when it does that, as soon as it does that conjugate addition, it'll the molecule actually attack that CH2 that's on that nitrogen containing methylated compound. Once you get there, the process, essentially you've now added a methyl to it, and what usually happens is then you get a deprotonation that's adjacent to that methylated bit there, and then it'll release the enzyme. Okay, so if you're able to, and ultimately sort of on the right side, you ultimately end up with a methyl there by sort of a deprotonation reaction to the nitrogen. But ultimately what you get then is it's not methylated. So you can imagine if you now put a substituent there that doesn't leave, then you're going to cause sort of suicide inhibition of this molecule. I don't know if it's on the next slide, but ultimately what people have done is they put sort of a fluorine there, so that fluorine can't be taken off right in that X position, and so that entire reaction mechanism is halted. Really, really cool design. It doesn't work as well with other constituents from what I understand, but nevertheless, the fluorinated compound has been really, really good. I think what we're going to do, we're going to leave it there. We're going to talk a little bit about ribonucleotide reductase at the beginning of next class, and we should wrap up anti-cancer section next time.